In this video, we will discuss about cognitive effect of CAR T cell therapy. And in order to check this, they use three different patient-derived xenograft model. First one is the cancer models belongs to CNS, that is central nervous system. Cancer model does not belong to CNS. And third one is cancer model does not belong to CNS in immunocompetent mice models. And thereafter, they check the tumor clearance by injecting these CAR T cells at different conditions. First, they use CAR T cells that can target GD2, and in this case they use this cancer cell lines, this diffuse intrinsic pontine glioma, and they can see that CAR T cells has an effect in clearing the tumors. Next, they use the CD19 CAR T cells, it is acute lymphoblastic leukemia. So in this case, they can see that the clearing is also occurred comparatively faster compared to the mock. Thereafter, they use the aggressive osteocarcinoma. In this case, the CD19s are expressed on the this osteosarcoma, and here we see the clearance is much more faster. And after that, they use rapid clearing osteocarcinoma. In this case, they use the GD2 CAR T cells, and here also they see the clearing is occurs much more faster. So here they not only use GD2 CAR T cells, they use CD19 CAR T cells also, and here also we can see the clearance is comparable to the on target, it is GD2 CAR T cells, and they also use the BH3 CAR T cells. So all the three results seems to be similar. Thereafter, they use the murine melanomas models where they use the CD19 CAR T cells, and here also we can see that the clearance is much better compared to the mock treatment. Thereafter, in order to check the cognitive effect, they use this NORT or novel object recognition test. So in this case, mice needs to identify the novel object. So when they did this NORT on this diffuse intrinsic pontine glioma, they can see that compared to the mock T cells controlled and the CD19 off target CAR T cells, they do not see any significant difference in this NORA test results. But when they use GD2 CAR T cells, they see a decrease in the recognitions. And similar results they see for acute lymphoblastic leukemia and this aggressive osteocarcinoma. So this indicates that is the clearance of tumors using CAR T cells might have some impact on these cognitive behaviors. Thereafter, they did, but the interesting thing that they find out in the case of rapid clearing osteocarcinoma, they do not see any significant difference. And similarly, when they use the chemotherapies, they do not see any difference compared to the vehicles. And when they use CD19s for this murine melanoma, they also do not see any significant difference compared to the mock T cells. Mock T cells is used as a control. So these two results are somewhat like does not follow the trends. So thereafter, they also did another test studies, that is T-maze test. So in this case, the T-maze test is the popularly known test to check the cognitive studies. So the results shows that the percentage attentions goes down. Percent attentions is lower even when the mock T cells controls are used, but when it's compared, or when the percentage attention is compared for these GD2 CAR T cells on target therapeutic, we can see a further reduction in the percentage attentions. And similar results we have seen for this acute lymphoblastic leukemia cells T-maze test, and also for this aggressive osteocarcinoma. But for the case of rapid clearing osteocarcinoma, we do not see any significant difference both for the on-target and off-target therapeutics. And similarly, for this, the case of T-MACE test for the chemotherapy, we do not see any significant difference. But for this NORA test, we see a significant difference. So in together, all of them indicates towards the point that cognitive effect will get affected by the CAR T-cell therapies in most of the cases. Cognitive effect can be seen in the patients who are treated with CAR T-cell therapies in most of the cases. This is the right statement that comes out from these results. So if you need to understand more about their experimental details in depth, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.